My name is Dylan Shuwan, and I will be presenting my presentation for human genetics, where we will be understanding the phenomenon of hair color change during puberty. A little bit of background information. I was curious because in my family, particularly my dad's side, there have been moments where individuals are born, they have blonde hair, but upon the age of puberty, that hair color changes to a dark brown. There's not a lot of research in this phenomenon, so I had to really investigate what may be the cause. I found that there is a family of polygenetic hair color genes that are expressed and change over time. M1CR is one of those genes. MC1R actually controls a red hair phenotype. I will touch on this later because of the nature of this phenotype, yet the fact that it has a high penetrance in blonde haired groups. However, the M1CR gene is well known to control the protein expression of tyrosinase, which is a primary protein in pathways for dark hair color colorization. Additionally, it has been shown that this protein is affected by hormones produced on the onset of puberty. The significance of understanding this phenomenon will provide a deeper understanding of the genetics of my family and may explain some of the mechanics of gene expression in hair color. Again, this phenomenon is not well documented, and I had to draw my own conclusions. Therefore, I hypothesize that a mutated M1CR gene may induce the expression of the protein tyrosinase at different levels when paired with hormones expressed in puberty by altering the expression of the alpha MSH ligand. In the multiple papers that I reviewed, there was particularly mice studies and human studies. In the mice studies, there was insertions of alpha MSH ligand, various analyzations of protein expression over age groups, and they used Northern blots to verify those expressions. In human studies, they used GWAS studies in large populations of prepubescent children and PCR to verify those expressions. They also used what's called the Fisher-Sellers descriptive scale to normalize hair color ratings in their data. To begin, we will be looking at the Grinchwald 1 1966 paper, particularly figure 1. They went ahead and mimicked the overexpression of the M1CR by injection of its target ligand. They injected alpha MSH, and what they found was a hair coat color change from light to dark. Now, again, what is MC1R's role in this? It controls the protein expression of the melanocortin 1 receptor, which binds to the alpha MSH ligand. Alpha MSH promotes the melanogenous pathway in melanocytes, which produce three key enzymes, one of which is tyrosinase. This is the building block to my hypothesis, suggesting that MC1R, particularly a mutated MC1R, may be responsible for the changes in pathways later which will alter hair color. Additionally, I had to look for a correlation in why it might change in puberty. In the Thrody AJ et al. 1922 paper, figure 1, they showed that in C3H heavy mice, a general mice strain, that coat color changes through the during pubertal stages. This suggests that gene expression may be affected by sex hormones. Now, looking at the tyrosine changes expression in mice during puberty was continued in that paper. They went ahead and determined expressions of proteins during that time period. They found particularly that the protein tyrosinase, one of the key proteins expressed downstream of MC1R, was increased in neonatal and pubertal mice. They found in yellow-coated mice that the hair color darkened when tyrosin expressed by nearly twofold. This was verified in figure 3, and shown quantifiably in Figure 2. Now, we look at some human studies in the CTEC A. et al. 2016 paper. They performed analysis on prepubertal children on pigmentation color genes by using PCR. What is interesting, again, looking back at our knowledge that MC1R is connected to a red hair color phenotype, yet its highest penetrance was found in blonde colored groups. Now, this is why I suggest that MC1R may be mutated. A normal MC1R gene would induce a red hair color change, particularly under maybe other circumstances, yet its high penetrance in blonde groups and with earlier data suggests that maybe mutation may arise in blonde-haired groups, and upon puberty, that will change expression in brown to a brown color. Additionally, the paper also did acknowledge that sex hormones may alter the expression of these genes. One thing I want to also note is that this study was done on prepubertal children. No studies were done further on same groups later after puberty, so I have no data to determine from these studies if any of the children expressing these genes did in fact change hair color. But then again, perhaps the children in these studies may have not had a mutated MC1R gene. Regardless, I decided to put together a suggested model as to why the M1CR gene may be responsible. Looking at a prepubertal stage, which would also represent a normal M1CR gene that is not mutated, it would have downstream of it normal alpha MSH levels, a normal melanogenesis pathway, and normal tyrosine levels, thus a blonde or light hair coloring. However, for a mutated M1CR gene during post-puberty, sex hormones go ahead and affect it, which causes an elevated change in alpha MSH levels, which alters the melanogenesis pathway, elevates tyrosine levels, which may cause a brown dark hair coloring change. So, conclusion and future direction. Once again, I must state, there is not much research on this phenomenon, at least in humans. However, the connection of dark hair color change response to increased tyrosine expression in mice, both by insertion and during puberty studies, suggests that an overexpression of the protein is sufficient to induce color change to a dark phenotype. Additionally, it suggests that the sex hormones play a role in causing this change. M1CR was shown to have its highest penetrance in prepubertal children that express blonde or dirty blonde hair, similar to my family, despite the fact that it is well known to cause a red hair color phenotype, or at least correlated with it. I suggest that there may be a connection with the mutant M1CR gene and alter expression when exposed to sex hormones. Future direction would be to identify other polymorphisms of the MC1R gene and determine expression in response to sex hormones during puberty and expression in downstream pathways. This would additionally also explain why, despite its correlation with a red hair phenotype, it doesn't always, high penetrance doesn't always correlate with red hair colored children or phenotype. These are my references for my presentation. Thank you for listening.